शुरू हो गया सर, राइट सर? So no problem, start.
Yeah. So from stem cell to organ building, what we are thinking that this will be a final solution and we won't have to take the drugs as such, and the stem cells and especially the autologous stem cells will help us in our treatment. Now, every 30 seconds, someone dies due to diseases in an organ that could be regenerated by stem cells or tissue regeneration, because so many accidents occur and we lose our organs or some of those things. And tissue in us do regenerate, but largely for the purpose of wear and tear. We know the liver has a potential to grow up to some extent, and even bone marrow is continuously growing. So biggest question, how to get back functioning organ in case of a degenerative diseases as Alzheimer disease or even tumors, how we can take up. Now, regeneration very much possible in plants. We see that because they're totipotent, and celery, see how it grows after 6 days. We know that way. And we also know about a bionic man, how it works. And we had been thinking about those things, but now they are coming reality. Because like plants, in animals also, regeneration existed. And we know salamanders, and if the tail is cut, it can regenerate itself. And somehow this will not work. Now, see, and we are neither plants nor machines or the reptiles. Then how then is it possible? That's the need of the hour. कि ना तो हम पौधे हैं, ना मशीन्स हैं, बायोनिक मैन आई टोल्ड यू, और ना हम रेप्टाइल्स हैं. तो रेप्टाइल्स में रीजनरेशन पॉसिबल है, पर हमारे नहीं. तो how can we do that one? That's a need of the hour, specifically for the cure of degenerative diseases, cancers, a huge breakthrough that could save millions of lives worldwide. Now, examples to show difference between regeneration and repair, because we know that many times when the cut is there or some organ is broken, it gets repaired itself, but it doesn't get regeneration, except for liver and bone marrow and some others, where regeneration can take place to some extent. Now, see, this is what normal hepatic lobules, and this how when injury occurs, and how the injury to cells and then its cells and matrix that takes place over there. Now, how could this occur? This is because the types of cells and processes these follow. See, we know that the stem cells. Now, we don't call them the root cells. Stem cells mean they have come out from something. जैसे जड़ से तना बनता है, that's why we call it stem cell. They have come out from something, but they remain as such in the body, and they can divide, but they cannot differentiate unless some signal is there. So we have a stem cell based populations. They may die sometime because if they success, or they may proliferate, or they get differentiated, and it depends upon the circumstances, and that is how the population is maintained. And we know that when skin is cut and it gets repaired, because the stem cells underneath, they come up, they start dividing, and up to some extent they divide, but half of them utilized for the repair and half are retained over there as. Now, there are three types of cells: labile cells, stable cells, and permanent cells. As the name indicate that what is their function. Labile cells have a high rate of loss and replacement, and therefore high capacity for regeneration. And common example is our mouth cavity. हम क्या पत्थर छोले सब खा लेते हैं, but अगर ये जितना छिल जाता है और our uterus or even the whole of the this nose and the cells go, but they come up again, and this is because the cells lose and they get replaced, and they high capacity of regeneration, such as squamous cells and glandular epithelia, hemopoietic cells in the bone marrow, and there's some stable cells. They do not normally proliferate, but can be stimulated to do so after damage, such as renal tubular cells, hepatocytes, osteoblasts, endothelial cells, and fibroblasts. Now, these cells normally do not proliferate unless some signal comes, and that comes from the some damage or some injury. And there's some permanent cells. They are unable to divide after initial development and therefore cannot regenerate when some are lost, such as neurons, skeletal and cardiac muscles. But let me tell you, now people have been able to now to grow the neurons by reprogramming, and even skeletal cells being reprogrammed, and possibly in the years to come, time to come, we'll be able to grow neurons in the lab also. Now, the key to all these cell types is stem cells, which have revolutionized the field of medicine and which are the futuristic therapy, what we call today regenerative medicine. There's two types, cell therapies, other are tissue engineering. Now, cell therapies, when only cells are involved in making a tissue or just repairing some part of that one. But tissue, when some organ is lost, some tissue is lost, how that can be regenerated? If supposedly trachea is damaged, how that can be replaced? That's a tissue engineering. So cell therapies and tissue engineering. Now in cell therapies, we have stem cells, or we induce the regeneration. That is the stable cells which are not able to divide themselves, but we can induce the regeneration. We know certain the factors which can be done. And in tissue engineering, what we required, जैसे कोई मूर्ति बनानी होती है, उस मूर्ति बनाने के लिए कुछ सांचा चाहिए होता है, तो उसी तरह इसमें स्कैफोल्ड यूज होते हैं. Bioscaffolds are used for whole organ scaffold, so that transplantation of in vitro grown organs and tissues, and also nowadays there's a bioprinting of organs. ऐसे प्रिंटिंग करते हैं हम प्रिंटर पे, पेपर पे डायग्राम्स की, इसी तरह नाउ bioprinting of organs is becoming possible, and certain machines have come up. So that is the way the things are moving in parallel. So what the stem cells are now? Now, what are stem cells? Stem cells are the raw material from which all of the body's mature, differentiated cells are made. Stem cells give rise to brain cells, nerve cells, heart cells, pancreatic cells, etc. ये वैसे ही है कि जैसे हम कुछ पैसे बैंक में जमा रखते हैं, तो बॉडी में भी हर ऑर्गन में ये कुछ स्टेम सेल्स पड़े रहते हैं कि जो अपने आप को अलाइव रखते हैं. कैसे रखते हैं? कि उसके साथ में जो डेवलप्ड सेल्स हैं, डिफरेंशिएटेड सेल्स हैं, वो इनको मेंटेन रखने में मदद करते हैं, क्योंकि ये इनका घर हो जाता है, इनका होम हो जाता है. बट देयर इज अ सिग्नल, देयर इज अ टॉक बिटवीन द टू, द डिफरेंशिएटेड सेल्स एंड द स्टेम सेल्स, दे टॉक विद ईच अदर कि आप, स्टेम सेल कहता है कि मुझे जीने दो, और जब आपको जरूरत पड़ेगी तो मैं आपकी मदद करूंगा. और वो जो डिफरेंशिएटेड सेल है, वो ये कहता है कि तुम जियो, मैं आपको खाना भी दूंगा, सब कुछ दूंगा, पर मेरे काम में इंटरफियर मत करना, जब तक मैं आपको इशारा ना करूं तब मेरी मदद करना. This is how it's a beautiful phenomenon in all our organs, wherein the stem cells are lying as a reserved money in the bank of the organ, and they are, जब हमें जरूरत पड़ती है हम बैंक से पैसा निकाल लेते हैं. तो उसी तरीके जैसे कि इनफैक्ट जैसे इंटरेस्ट निकालते हैं, वैसे हम स्टेम सेल निकाल लेते हैं, जो वहाँ पे कुछ पड़े रहते हैं और कुछ बार ऐड हो जाते हैं. सो दिस इज a beautiful story, like a social, we know the social life also works like that. So there are level of organizations in the body, all of us know. There are cells, tissue, organs, and organ system. Yes, we have the molecular structure of cell and all those things. And we are not referring to that one because nowadays the things have gone up to the even the molecular level for that purpose. But see, with these cells, they together form a tissue. It's not a one type, but many type of cells form a tissue, and the different type of tissues join together to form an organ, and the organ by makes the organ systems for respiratory system, digestive system, and all those circulatory system are formed, and all these organ systems make a body of the person. Now, these stem cells have certain general properties. That is unspecialized cells. They are not specialized. They are simple, common cells capable of dividing themselves for long periods. That is what called self-renew. They can renew themselves so that क्योंकि एजिंग से कुछ सेल मरते भी रहते हैं तो उसका रिन्यू होता रहता है कि बैंक में पैसे कम मत हो, बैंक में सेल कम मत हो, स्किन के नीचे सेल कम मत हो, तो सेल्फ रिन्यू करते रहते हैं. बट जब जरूरत पड़ती है देन गिव राइज टू स्पेशलाइज्ड सेल टाइप्स कि वो डिफरेंट जैसे लिवर में हेपेटोसाइट्स बनते हैं और दूसरे बनते हैं तो बनते रहते हैं. जैसे दी स्टेम सेल्स दे हैव सेल्फ रिन्यूअल, अपना साइकिल चलता रहता है. जब जरूरत पड़ेगी डिफ्रेंशिएट करेंगे और मैच्योर सेल बन जाएंगे. देन इट इज पॉसिबल बिकॉज these cells have a different capacities. और ये एक नई चीज नहीं है, हमारे भारतीय सभ्यता बहुत पुरानी है और ये जो हम बात आज करते हैं ये बहुत पुरानी है. इवन सुश्रुता had been doing that one. जो प्लास्टिक सर्जरी है वो दुनिया को हिंदुस्तान ने दी थी. The first plastic surgery was done by Sushruta, what we call a cosmetic surgery, since technique for repairing the disfigured nose with a flap of skin from the forehead in practice almost unchanged in technique, what we call rhinoplasty to this day. So this is the Indian culture. Whether you talk about mathematics, you talk about biology, you talk about certain chemistry, physics, we were very strong. Indian culture had been a scientific culture. आज जो हमारे त्योहार हैं वो भी साइंस बेस्ड थे, हमारे रिचुअल्स हैं वो साइंस बेस्ड थे. जब हवन है कुछ भी चीजें हैं सबका साइंटिफिक बेसिस है. पर आज हम क्या करते हैं, हवन करते हैं तो वो एक मौली का धागा दे के कहते हैं वस्त्र समर्पण. अरे उसका सब चीजों का मतलब था, या फ्रूट देना उसका मतलब था पर हम भूल गए. या इवन तिलक लगाना माथे पे है उसका भी मतलब था. या लेडी जो पाँव में एक वो सिल्वर का डालती थी उंगल में एक रिंग सी, उसका भी मतलब होता. मतलब हमारे जितने भी रिचुअल्स हैं इंडियन रिचुअल्स दे आर साइंस बेस्ड. हम भूल गए. बट नाउ आई थिंक टाइम हैज कम, वी हैव टू रिन्यू अवर ओल्ड साइंस in a new language or new technology. So Canadian researcher Ernest M. McLeod and James in 1963 first description of self-renewing activities of transplanted mouse bone marrow cells. In 1981, Martin Evans and Matthew Andrew Kaufman, mouse embryonic stem cells, Department of Genetics, University of Cambridge. And 1998, James O. Thompson, University of Wisconsin-Madison, removed cells from his pair embryos at fertility clinics for in vitro culture, brought stem cell research into limelight, established the world's first embryonic stem cell line, which still exists today. That's the two main types of stem cells, embryonic stem cells and adult stem cells. Now, embryonic stem cells are there when the embryo is there. At that time, we get the certain cells over there which can be made to differentiate in different type of cells. And adult stem cells, which almost all the organs have the bank of the stem cells. And special attention is to umbilical cord blood stem cells, induced pluripotent cells, and stress-triggered acquisition of pluripotency, what we call STAP, but it is a controversial, and that's why not much progress has made this one. Now, what are the embryonic stem cells? Now, when natural fertilization takes place, the zygote divides into two, then four, then eight, and then it may be a blastula. But inside the blastula, there is a group of cells, mass of cells, which are almost totipotent, rather pluripotent, and they can mate to differentiate into different type, where they can be grown in. And this is possible in vitro fertilization, which is common because normally when in vitro fertilization is done, it's a more than one egg gets fertilized, and we can implant only one fertilized egg into the body of the womb of the woman. The rest can be frozen and they can be used if they need in the future. But we may have to spoil them, throw them away after some time. Now, instead of doing that, if we can keep them and use them for growing certain tissues or organs, that will be that facilitate their therapy progress. But people are not agreeing. They say no, zygote itself is a life and we should not destroy it. There's a controversy. There's different schools are going on of thought which they discuss. Ovum that has had a nucleus removed and nuclear material injected from intended recipient of final tissue product, that is reproductive therapeutic cloning, is that we can have a ovum because women every month produce the ovum, and if that ovum is taken, its a nucleus is removed, and we take some nucleus from some other cell and do that way, the things can be possible. Now see, in embryogenesis, we know day one fertilized egg, day two cells are formed, then four are formed, then 8 cells form, and this is what we call totipotent stage, that these cells can be made to form any type of cell. Like a plant cell, they can be utilized. Then from there, after 3-4 days, we get a sort of a ball of cells, a multi-cell embryo is formed, which has a blastocyst outside, and then we have this group of cells, mass of cell, and that can differentiate from different type of tissues. If these cells are removed, then they can be cultured and used for the growing of the certain tissues, etc. And this is how after 9 months, his child is born. But this, up to this, things are possible. Then as we move down, the differentiation level goes on decreasing. The utilization potential of differentiation goes on decreasing. And that's how they have been named as such. Now, embryonic stem cells are very controversial, and we have to find out the alternative sources because people don't say that the embryo is a life itself. Even a zygote, life starts, and we can, if we spoil it, it will be a murder. Or even if a blastula, we try to take out the cells, blastula itself will die, and that means we are killing the person. But the second school says, no, we have to throw that in the garbage, that embryo, and we're taking out a cell for treating more than one person, it will be useful. It will be useful for the society. So now then there are somatic cell nuclear transfer, what we call as SCNT clone, that we can take a human egg, we can take a somatic cell, and from the human egg, we can take out the nucleus, then it can be fused with that, and after fusion, we can form a blastocyst, and then it can go. So embryo develops, or we destroy the embryo. So this is therapeutic cloning. Now, so that because it is possible, but we are not pursuing much unless or until the controversies go. And most of the work is being done on adult stem cells because they are multipotent stem cells. That the brain has it, heart has it, bone marrow has it. So the brain one they can form their neuronal cells are formed from the heart cells. We have the cardiac muscles and other cells are formed from bone marrow. We know different type of blood cells are formed. They are regularly forming, and we can exploit their potential for our pulse cell. So stem cell derived from brain, heart, and bone marrow give rise to cells of that very particular tissue. And this is how they are in every organ, everywhere we have the stem cells, and they are in association with the normal cells. That is how they survive. कि जियो और मुझे जीने दो. The stem cell को वो बॉडी सेल कहता है कि आप जियो, नेबरहुड सेल कहता है आप जियो पर मुझे जीने दो. ये फिनोमिनन जो हमारा सोशल लाइफ में है वो इसमें है बिकॉज थ्रू सम ऑफ दीज signals they talk to each other. They are all proteins, so factors. They talk to each other. Stem cell and the differentiated cell, they talk to each other, and they help each other when a time is required, when they are needed. They help. So in every organ, we find that these stem cells are there, and there's a homing. A lovely home is there, a lovely neighborhood is there, and once the neighborhood is destroyed, stem cell will not work. That is a problem, and you know that अगर हमारा neighborhood अच्छा नहीं है तो हमारी जिंदगी भी खराब हो जाती है. इसी तरह अगर stem cell के neighborhood अच्छा नहीं है या normal cell को neighborhood अच्छा नहीं मिला तो destroy हो जाती है. So adult stem cells are, the problem is there. It's not so easy that they're aging. With the see, we know that they don't do anything, but still by staying over there, each cell produce so many things, some proteins, etc., attached to the wall of this, and which create aging of that one. Of course, the epigenetic also plays a role in that. And secondly, the yield is too low. It's not much in number that you can grow the tissue as such, and they are mostly multipotent. Then umbilical cord blood stem cells, they are very useful because they anyhow we have to throw them. When a child is born, you cut the umbilical cord and you throw that one, but that can be stored, and we have now cord banking, cord stem cell banking, and many people in foreign countries are doing that one. That some this umbilical cord banking and cord stem people are themselves from their own futuristic child, अगर child को गड़बड़ होती है तो उसके लिए stem cell store रखती हैं. Now, induced pluripotent stem cell, that cells को induce किया जाए और वो अपने organ बना दे, तो इस पर Nobel Prize in Physiology given in Medicine 2012 jointly to Sir B. Gordon and Shinya Yamanaka for the discovery that mature cells can be reprogrammed to become pluripotent. And then Yamanaka factors are a set of 4 transcription factors. When introduced into cells, induces stem cell formation. कि जो differentiated cell है, अगर उसमें कोई factor डाल दिया जाए, तो वो reprogram हो जाता है. Because what is the differentiation? There are multi-step stage process. और अगर उसको reverse कर दिया जाए, तो वो अपना normal stem cell बन जाता है. And there is a lot of success in that one. People have been able to reprogram that, and it's how Yamanaka factors, 4 transcription factors is discovered, and which introduce cells, they take back the cell कि हम दिल्ली पहुंच गए, अब दिल्ली से वापस जबलपुर कैसे पहुंचना है वापस, तो वो रास्ता ये फैक्टर बता देते हैं. Then John Gurdon used nuclear transplantation and cloning to show that a nucleus of a differentiated somatic cell retains the totipotency necessary to form a whole organ. क्योंकि जो हमारा न्यूक्लियस होता है, चाहे वो लिवर सेल है, पर उसमें जीन तो वही रहेंगे जितने ओरिजिनल में थी, जो 25,000 चीजें कुछ भी हैं. Now if we know the pathway, तो वो जीन जो है क्योंकि स्विच ऑन स्विच ऑफ चलती रहती है, that's how cell differentiated into liver cell, stem cell, brain cell. तो जीन सभी में एक जैसी हैं, पर किसमें कौन सी जीन एक्टिव है वो डिपेंड करता है, वो नर्व सेल बनेगा, लिवर सेल बनेगा कि ब्रेन सेल बनेगा। अगर हमें ये फैक्टर पता है कि ये मेरी जीन जो है मसल सेल एक्टिव होने से मसल बनती है, तो हम जो बोन मेरो सेल है उसमें उस जीन को एक्टिव कर देंगे, एक्टिव करने से वो लिवर सेल बनाने शुरू कर देगा। सो दैट्स व्हाट वी कॉल रीप्रोग्रामिंग, कि गोइंग बैक टू इट्स पाथवे, क्योंकि द फॉरवर्ड पाथवे इज डिफरेंशिएशन, रीप्रोग्रामिंग इज अ बैकवर्ड पाथवे, कि जिस पाथ से आए थे उस पाथ से वापस जाना है। कि आपको दिल्ली पहुंचे थे, पहले रास्ता नहीं पता था, अगर दिल्ली से वापस जबलपुर जाना है तो आपको वो रास्ता याद रखना पड़ेगा, तभी वापस पहुंचोगे। ये ना हो कि आप बम्बई पहुंचे। दैट्स इज द रीप्रोग्रामिंग ऑफ द सेल्स व्हिच इज गोइंग ऑन नाउ। Induced pluripotent stem cells, that is, we take the fibroblast and put them, these four factors are put in that one, then they form the colonies, and these four colonies then can be made to make cardiomyocytes, endothelial cells, smooth muscle cells, neurons, red blood cells, adipocyte. Each one requires a specific factor for doing that one. And in fact, it's not new. When I was there in 1999 in a plant genomic research laboratory in Germany, the erstwhile East Germany, I was in a. Then the director took me to his wife's lab, and she was working on the animals, in the mouse cells. And they took me to the lab, and they showed me in a one petri dish, the cells are growing in three type of cells: the neuronal cells, cardiac cells, and muscle cells. Mean in one petri dish, they could differentiate the three different type of heart cells in 1999. And not like India, कि अगर बॉटनी में काम करते हो बॉटनी ही करनी है आपने, zoology नहीं कर सकते। वहाँ पे प्लांट जीनोमिक रिसर्च लेबोरेटरी में वो एनिमल पे काम चल रहा था। एंड दैट हाउ द थिंग्स, देयर इज अ लॉट ऑफ इंटरेक्शन between different sciences, and rather now we are talking about integrated science, or what we today call genomics, and it's all because of that. Cells, they can reprogram, as I told you, donors, we can take a donor somatic cells and they can reprogram to form different type of cells, and supposedly a person requires a particular tissue. What can be done? क्योंकि इम्यून रिजेक्शन हो जाती है अगर बाहर से किसी और के सेल लोगे, तो उसी आदमी का डोनर सोमैटिक सेल लेके उसको रीप्रोग्रामिंग करके और उससे उसके सेल बना के वापस उस आदमी में डाला जा सकता है। व्हाट वी कॉल द ऑटोलोगस सेल थेरेपी। अगर दूसरों में डालोगे तो एलोजेनिक कहेंगे, एलोजेनिक में रिजेक्शन हो जाता है, इम्यून सिस्टम में रिजेक्टेड, बट ऑटोलोगस में वो रिजेक्शन नहीं होती। यही सेल जीन थेरेपी के लिए यूज हो सकती है कि एक सेल में अगर हमने कोई जीन म्यूटेटेड है या उसको ऑगुमेंट करना है, कम प्रोटीन पैदा करता है, ऑगुमेंट करना है, तो उसके लिए जीन थेरेपी की जा सकती है। और अब तो जीन एडिटिंग इज आल्सो पॉसिबल। क्रिस्पर टेक्नोलॉजी हैज अलाउड टू इवन व्हाट वी कॉल द जीन एडिटिंग, कि कुछ को निकाल दीजिए। इवन so many things are now possible. I'm not going to take that one. So now we have the stem cell. Now how to engineer them to form that tissues, which is a very hot topic now. Combine stem cell potential with scaffolds to engineer organ. What is a scaffold? Scaffold basically एक सांचा है जो उसको वैसा ही ढांचेगा। अगर आप कोई मूर्ति बना रहे हो राम की, कृष्ण की, या कोई एनिमल बनाते हो, तो उसका जो कुम्हार है वो सांचे रखे बैठा है, उस सांचे से बनाता है। तो इसी तरह अगर हमने किडनी बनाना है, हमें उसका वैसा ही सांचा चाहिए जिसको हम स्कैफोल्ड बोलते हैं। Now scaffold can be this plastic or other materials have come up, nanomaterials, so many materials are there, but they can create a problem. Now what we also did here sometime, that we took the rat kidney, removed the cells from within the kidney, and make that's like a empty bladder, and then we re-injected normal cells into that and tried to differentiate that into the renal cells, and we got a quite success. So this is the field we call a tissue engineering, a field that evolved from the field of biomaterials development, wherein the scaffolds, cells, and biologically active molecules were combined into functional tissues. It entailed assembling functional constructs that restore, maintain, or improve damaged tissues or whole organs. Some of the most common engineered products are approved by the in America, the Food and Drug Agency, artificial skin and cartilage with a limited use in human patients. So we have so many human organs, and a trial is being done in almost all these organs. But we have a success in the case of trachea, which has been this is a great success, and they have been replaced in some of the children also. Now, the process tissue engineering promise through the following info. Cells are the building blocks of tissue, and tissues are the basic unit. Groups of cells make the secrete their own support structures called extracellular matrix. The matrix scaffold beside lending support to these cells also act as a relay station for various signaling molecules. These cells receive messages from many sources that become available from the local environment. Each signal can start a chain of responses that determine what happens to this cell. By understanding how individual cells respond to signals, interact with their environment, and organize into tissue and organisms, where researchers have been able to manipulate these processes to mend damaged tissues or even create new ones. So this is a complex mechanism which involves in both macro and micro environment and the materials. So when we have the macro and micro environment, material is there. The only what is required, we have to use a technology how to assemble them and to make them to interact with each other, because the signals have to be passed on from one component to the other. The process often begins with the building of scaffold from a wide set of possible sources, from proteins to plastics. Once scaffolds are created, cells with or without a cocktail of growth factors can be introduced. If the environment is right, a tissue develops. In some cases, the cells, scaffold, and growth factors are all mixed together at once, allowing the tissue to self-assemble. Another method is to create new tissues using an existing scaffold. The cells of the donor organ are stripped, and the remaining collagen scaffold is used to grow new tissue. The process has been used to bioengineer heart, liver, lung, and kidney. We also have got some success in kidney. This approach holds great promise for using scaffolding from human tissue discarded during surgery and combining it with a patient's own cells to make a customized organs that would not be rejected by the immune system, what we call autologous system. Now, currently tissue engineering plays a relatively small role in patient treatment. Supplemental bladders, small arteries, skin grafts, cartilage, and a full trachea have been implanted in patients, but the procedures are still experimental and very costly. While more complex organ tissue like heart, lung, and liver have been successfully recreated in the lab, still long way from being fully reproducible and ready to be implantable. These tissue, however, can be quite useful in research and especially in the drug development, because you can check the drug whether the person is a because every drug doesn't save every patient. It may not, every patient may not respond to the same drug. Even we don't respond similarly to the paracetamol, what we call a personalized drugs. And now in vitro techniques are coming up by using these cells, and patient's own cells are taken out, cultured in the laboratory, drug is given, what we call colony forming assay, and then we see how these cells are responding to the drug, and if they are responding well, then drug is given, otherwise drug is rejected. Now you can use the multiple drugs for a multiple type of cells and do that one, and you can, there's a scanning of the drugs in vitro and then give it to the patient. Then patient will not reject and he will be healed. Using functioning human tissue to help screen medication candidates could speed up development and provide key tools for facilitating what we today call personalized medicine, while saving money and reducing the number of animals used for research, because you know that we can't use animals now for most of the research, but here by this we can reduce the number. Now these have been working exemplified naturally. You know that skin shedding and tissue regeneration in African spiny mouse, which will take place, and hawk hairstyle has been done, and this is how people have grown ear over there. Now, stem cell based tissue engineering take care replacement in a child, a 2-year follow-up study was done, and the child could survive, and even after 2 years. So this is how Dari Warren and Lee Yang visit their 2-year-old daughter, Hannah Warren, in a post-operative area children's hospital in Illinois, having received a new windpipe in a landmark transplant operation which took place in 2013. Now, many tracheal transplantations are taking place. Now, and one own stem cell to rebuild the airway in his body. His own cells were used to rebuild the airways. This 11-year-old boy. Then in India too, India is not lagging behind. LV Prasad Eye Institute, Hyderabad, has performed over 900 stem cell based reconstruction of damaged corneal surface, and they are doing wonderful job under the leadership of Dr. Bala Subramaniam. Now, now we have know that epicell cultured epidermal autografts, integra skin graft, and dermagraft skin took place in 1995. So this skin replacement is very common. And then we have a bioartificial liver device, artificial pancreas, artificial bladders, cartilage, scaffold-free cartilage, and tissue-engineered blood vessels. They are all in the pipeline, and some have been used experiment. Now, organ engineered in the laboratory, urinary bladder, ear, lung, tooth. A lot of work is being done on the teeth, let me tell you. Then kidney, heart, and now mice with humanized livers for drug screening are being used. As I told, in vitro, now in vivo also, this is possible that you can take the cells from the human liver, and they can put in the nude mice or mice, knockout mice, and then grow them and check the drug effect of drug. And this is possible. What we call human liver cells organoid to be placed in a mice for drug screening. Now, major hurdle to tissue implant for engineering organ immunorejection solution lies in the gene editing technologies, and I already mentioned that now we can edit the genes which are responsible for rejecting the tissue, and then make the tissue to grow, and can be used that way. And now we have a very precise technology, CRISPR technology. Now underlines the potential worth 1.2 billion in 2012. It's growing to 30%, expected to reach around 16 billion by 2000, which has already done. The Indian market is to reach 600 million now. It is uncharted territory, and as science advances, new segments will be created, and now we know that this market will grow a lot. So stem cell technology can cut pharma R&D cost by 25% a year and save 225 million in each drug market, according to Professor Colin McGinn of the Newcastle Center of the Cord Blood. So latest revolution is there. Now bioprinting has come. Now you don't have to use the test tubes. You know that which type of cells form the liver. You put them in the test tubes and then give it direct in the through the computer, give a signal. Software programs are there, and they will print the organ. They'll make the organ accordingly, because these cells will be distributed accordingly as in direction given to the computer. So what we bioprinting is a new emerging technology that aims at achieving to develop new tissues and eventually organs. It is in the research phase, as with any technology that takes time to completely develop. Currently, there's a lot of research being done on bioprinters. In bioprinting, devices are printed by feeding biological material as deposits with an inkjet printer instead of ink. Cells are printed or deposited on a surface layer by layer, and in this way, full organ can be produced. So bioprinting aims at organizing the elements of the tissue during the fabrication step individually through the process of depositing layers of बायोलॉजिकली इंपॉर्टेंट कंपोनेंट्स. सो जैसे हम फिगर्स बनाते हैं, प्रिंटिंग करते हैं, आर्ट करते हैं, वैसे ही now organs are possible over there. And organovo holding creator and manufacture functional three-dimensional human tissues for medical research and therapeutic application presented at the 2013 Experimental Biology Conference in Boston. And since then, many new machines have come up with experiments are going on. So bioinks to be used 3D paper printer for printed scaffolds. We have a bioink materials, agarose, alginate, chitosan, collagen, decellular ECM, extracellular matricin, fibrin, fibrinogen, gelatin, graphene, so many which are biological materials, they can be used like this one. And we have but advantage disadvantage of each one of them. The trials are going on with this one. And then we have this in bioprinting, X2N based bioprinting, inkjet based bioprinting, laser assisted bioprinting, and there's no manual work. It's all computerized. What we now call it a integrated science, that is the what we call computational biology joining with the machine learning with a new biology. They all three are making the life going to make life more easier in the medicine area. We have tissue model cartilage has been done from bone, skin, blood vessels, which type of cell lines used, bioprinters used, bioink materials used, and the references are there for us. This is a so 3D printing scaffold skin for burnt patient. Skin is now, as I told you, the skin has been engineered and it can be used for healing the burning material. That's so 3D printing, heart, ear, cornea, blood, vessels, vascular network, coronary artery, nephron, etc. They have been prepared in the experimental stage. Now, innovations needed for engineer Indian bioprinter, need of the hour, step towards what we call Atmanirbhar Bharat, which is the dream of our Honorable Prime Minister. And I think days not far off. Indian brain is very sharp brain. Somehow we have not been showing our capacity in India, and we had been moving out. But now, since the facilities are being provided, we are encouraging the youth, and that youth can do wonders. And there is not far off when we will have an Indian bioprinter. The first bioprinter was made by Chinese. Now other countries have made it, but the Indian bioprinter, when it comes, the regeneration biology will be very, very easy. And I think it won't take much time. But experiments will ultimately reach into a success. Thank you very much, Dr. Sanjay, for this one.
Thank you, sir. It's a very nice presentation. It's a very nice presentation. Sir, thank you that on first day it was very difficult for us to broadcast this lecture, but today we have made it possible, and it's a very nice lecture. मैं सभी पार्टिसिपेंट्स को बोलना चाह रहा हूँ कि आज जिन्होंने ये बैठे होंगे यहाँ पर, वो एक आप घर बैठे आप ये समझ सकते हैं कि स्टेम सेल्स क्या है, टिश्यू इंजीनियरिंग क्या है. मैंने पहली बार देखा, हालांकि मैं भी साइंस कम्युनिकेशन करता हूं, तो साइंस कम्युनिकेटर जो होता है वो ही इज अ जैक ऑफ ऑल. मैं खुद पॉलीमर्स पे काम करता हूं, पॉलीमर फिजिक्स पे काम करता हूं, तो ये मैंने आज पहली बार अनुभव किया कि किस तरीके से टिश्यू इंजीनियरिंग से हम जो है इंकजेट प्रिंटर्स का यूज करके हम किस तरीके से जो है
बायोमेटेरियल पार्ट ऑफ दिस वन.
Yeah.
Your biomaterials are part of this one.
Yeah, sir.
It could not have been possible without your material, nanomaterials and all those things.
जी सर. वो ये तो मुझे ये हम लोग भी साथ में करते हैं, पर लेकिन इतना बढ़िया जो है, इतना अच्छा प्रेजेंटेशन जिसमें कि आपने सब कुछ बताया कि किस तरह से स्टेम सेल्स का नॉलेज, टिश्यू इंजीनियरिंग का नॉलेज जो है वो हम उसको सहायता से किस प्रकार से जो हमारे ऑर्गन्स खराब है, देखिए जानवरों में तो होता है, जानवर रीजनरेशन होता है, पर इस रीजनरेशन को हम मनुष्यों में भी कर पाए तो ये संभव है टिश्यू जो इंजीनियरिंग से और स्टेम सेल्स के नॉलेज से। तो एक बहुत ही सुंदर और बहुत ही अच्छा लेक्चर था। अगर पार्टिसिपेंट्स कुछ क्वेश्चन पूछना चाहें तो पूछ सकते हैं
या
और हम लेक्चर शुरू करेंगे। तो मैं इसको ऑफ नहीं कर रहा हूँ, शायद मुझे लगता है कि सर भी चले गए, मैं थैंक्स फिर से सर को कर रहा हूँ। हमारा जो लेक्चर सेकंड लेक्चर है वो 2 बजे का समय है, तो जैसे ही हमारे रिसोर्स पर्सन आएंगे तो हम इसको शुरू करेंगे, तो इसलिए मैं इसको ऑफ नहीं कर रहा हूँ इस मीटिंग को।
मैं कर दूं, मैं जरा लेक्चर चेंज
तो
कर
थैंक यू सर प्रोफेसर सोप्ती, और हम चाहते हैं कि आपका हमें आशीर्वाद लगातार मिलता रहे।
श्योर, एनी टाइम।
जी।
आपका
और
जिज्ञासा, ऑल द बेस्ट विशेस टू
जी,
जिज्ञासा।
आगे आगे जाके कभी अगर अवसर मिलेगा तो आपको हम जबलपुर में बुलाएंगे और होगा कोशिश करेंगे कि हम आपको लाइव सुन सकें, अगर ईश्वर ने साथ दिया तो बहुत अच्छा लेक्चर सर, थैंक यू
ये
सर।
साथ जल्दी मिल जाएगा सर, ये
मिलता
जल्दी
नहीं
मिल।
है सर साथ,
आई
साथ
नो
ये बहुत
पीपल,
कठिन काम है सर। मैं तो बोलता हूँ
पीपल आर
सर, जोगर
क्रिएटिंग
फूके आपने,
कंट्रोवर्सी।
आपने शायद सुना होगा ये कहावत, जोगर फूको आपने चले हमारे साथ, इसमें मिलता नहीं है। ये बहुत ही थैंकलेस जॉब है सर, ये बहुत कठिन है इसमें। तो
आई नो।
थैंक यू सर, थैंक यू सर।
नहीं, एक बात बताऊंगा, अभी डॉक्टर खन्ना बता रहे थे,
जी
कल
सर।
आर्टिकल मैंने भी पढ़ा है कि जी वो वैक्सीन यूज नहीं करनी चाहिए।
जी
अरे भैया, दिस इज अ पार्ट ऑफ द लाइफ। नाउ
जी
वैक्सीन जो है, इट वोंट इंटेग्रेट इन अवर बॉडी, इट वोंट इंटेग्रेट, it will counter the viral spike protein. It's not. And see, there are so many type of the mRNA vaccines are there, and people are developing it. I mean, people are trying to create a confusion. Of course, one apprehension is that the virus is highly mutable. कल ही आर्टिकल था कि 12,040 म्यूटेशन ढूंढी गई हैं, पर somehow none of these mutation has altered the cycle of the virus. पर अगर ऐसी हो गया तो वैक्सीन
Connected.
वैक्सीन
Sir,
फेल हो सकती है अगर ऐसी म्यूटेशन आ जाएगी तो।
no, sir. That was not my opinion. That was from Robert F.
या आई
Kennedy.
नो दैट, मैंने
Yeah,
आपने आपने पेपर की, मैं भी पेपर की बात कर रहा हूँ सर,
yeah, yes,
मैं पेपर की बात कर रहा हूँ। मैंने आपको कोट इसलिए किया कि आपने पेपर से दिया है, तो मैं
yeah,
भी उसी पेपर के ही रेफरेंस दे रहा हूँ
yeah, sir.
कि अनलेस ऐसे कंफ्यूजन क्रिएट करना, दिस इज नॉट, unless you are sure. Have you seen integration of that material into human cells? No. How can we just say that one?
सर, ये जो मैं बीच में आपसे वो कहना चाह रहा हूँ कि बहुत से कंफ्यूजन फैलाए गए हैं,
Yeah.
बहुत से कंफ्यूजन फैलाए गए हैं। वैक्सीन के लिए भी लगातार भारत सरकार भी इसके लिए कोशिश कर रही है, देख रही है कि कंफ्यूजन ना हो। अभी उन्होंने, मैं आज ही पढ़ रहा था कि उन्होंने एक अलग से ग्रुप बनाया हुआ है जिसका काम ही यही होगा कि रिलीजियस ग्रुप की भी मदद ली है कि कहीं ऐसा ना हो जैसे कि बहुत पहले पोलियो वैक्सीनेशन के साथ हुआ था और
Oh,
अभी भी पाकिस्तान में हो रहा था।
that's a,
तो बहुत सी समस्याएं हैं, तो रिलीजियस ग्रुप से भी उन्होंने मदद ली है कि जिससे कि अफवाहें और भ्रांतियों को दूर करें। देखिए,
yes,
मैं कल भी बता रहा था कि ये जो वैक्सीनेशन जो है, एक 5 साल 4 साल 5 साल चलने वाली डेवलप करने की प्रक्रिया 4 साल 5 साल लेती है, और इतनी जल्दी फास्ट ट्रैक काम हुआ है और बहुत अच्छी वैक्सीन आ रही हैं।
हाँ
जैसे
हाँ,
हाँ, स्पुतनिक
at
जो
least you
जिसकी
will
लोग
have at
मजाक उड़ा
least
रहे थे,
six vaccines
हाँ,
in the
और जो
next three to
स्पुतनिक की
four
सर जो
months.
मजाक उड़ा रहे थे, जो स्पुतनिक के लोग मजाक उड़ा रहे थे, उसकी 95% उसकी इफिकेसी है सर।
Yeah,
हाँ, स्पुतनिक की 95%। और एमआरएनए वैक्सीन तो निश्चित तौर पे गेम चेंजर होंगी। एमआरएनए वैक्सीन जितनी भी हैं, मॉडर्ना की, फाइजर के साथ थोड़ा सा टेम्परेचर की प्रॉब्लम है कि वो -80 डिग्री सेंटीग्रेड चाहता है, वो
that's a problem.
प्रॉब्लम है। पर लेकिन सर मॉडर्ना की वैक्सीन बहुत अच्छा है,
मॉडर्न अच्छी
92।
है, इवन आपकी ये जो ऑक्सफोर्ड वाली है,
ऑक्सफोर्ड की भी है, ऑक्सफोर्ड में
right,
आज
is
कुछ
also
थोड़ा
to
सा
work
उनका प्रॉब्लम आई है
in
उसका,
8 degree centigrade.
हाँ, टेस्टिंग में कुछ समस्या उनकी आई है, पर लेकिन वो भी बहुत अच्छी वैक्सीन है सर। तो वैक्सीन बहुत अच्छी आ रही हैं। इवन भारत बायोटेक का जो पुरानी टेक्नोलॉजी पे आधारित है, डेड वायरस पे लेके जो चल रहे हैं, वो
हाँ वो भी
भारत
है।
बायोटेक की, वो भी अच्छी वैक्सीन है, बहुत अच्छी वैक्सीन है। तो अभी सब लोग आशावान रहिए और
रहेगा
जब
सर।
तक के वैक्सीन नहीं आ रही तब तक के घर
सर,
में
वर्ड हैज टू
सुरक्षित है।
सर्वाइव,
हाँ,
वर्ड हैज टू सर्वाइव, वी मे नॉट सर्वाइव, वर्ड विल सर्वाइव।
और आज कोरोना है, कल को दूसरा कोई आ जाएगा।
वो तो आ ही रहा है, इट्स ऑलरेडी कमिंग।
हाँ, वो तो आता
If
रहेगा
you
नया।
look at the history of pandemics,
जी
it's a legal history of pandemics. It's a long history.
जी, बहुत।
And ये पिछले 10 साल में तो बहुत ही पेंडेमिक है अगर हम देखें तो।
और सर ये देखिए, हम लोग में से कोई भी ऐसा व्यक्ति नहीं है, इवन जो है जो आज जो राज कर रहे हैं अलग अलग देश में, किसी को भी अनुभव नहीं था इस पेंडेमिक का। भाई दो
दीदी,
1918 में आई थी पहले,
मैंने किताब
1918 में
लिखी,
पहले आई थी।
कोविड-19 पे किताब कंपाइल की है, साइंस, सोशल साइंस, इसका सब उसमें कंबाइन किया है
जी
कि अब ये जो वायरस है, इससे जो बाउंड्रीज एंड डेवलपिंग एंड अंडर डेवलप उड़ा दी है,
हम्म,
बेक उड़ गई है, अमीर गरीब की उड़ गई है। अब किसी लेबर को मरते देखा कोरोना से आपने,
जी
बिकॉज हिज इम्यून सिस्टम इज वेरी स्ट्रॉन्ग। वो छप्पड़ में नहाते हैं, हम तो बिसलेरी पानी पीते हैं, वो पानी पीते हैं नल के ऑर्डिनरी, उनको तो कोई कोरोना नहीं होता देखा मैंने।
जी जी जी।
इतना इसको शौक भाई, मेरा परिचय दो, मेरा परिचय दो, फिर उन्होंने कहा इनको पद्म भूषण मिला था, ये है, इतना शौक उस आदमी को। धीरे धीरे बोला मेरा पहले
हेलो,
परिचय
ये
तो
कौन
दे
बोल
दो,
रहा है?
पहले मेरा परिचय तो दे दो।
प्लीज शांत रहिए, जो भी है माइक बंद की हुई। अंजना
थैंक यू जी,
मैडम,
थैंक यू डॉक्टर साहब, थैंक यू।
थैंक यू सर, थैंक यू।
राइट, ऑब्लाइज्ड।
हम नेक्स्ट लेक्चर
ऑल
शुरू
द
करेंगे,
बेस्ट, ऑल द बेस्ट।
हमारे गेस्ट आ जाएंगे। थैंक यू सर, थैंक यू सर, थैंक यू प्रोफेसर सोप्ती, थैंक यू सर। नेक्स्ट सेशन हमारा चालू होगा, बस मैं इसको वो कर रहा हूँ, अभी लाइव में ही रहूंगा मैं। इसको